Okay, so now we'll come to the last topic in transcriptional control, okay, which is about transcriptional regulation through uh, chromatin modification as well as the mechanism of transcriptional repression. Okay, so far we have primarily focused on transcriptional activation. So now we will talk about transcriptional repression as well. Let us start with a very interesting phenomenon. So you know that yeast cells can undergo changes in the mating type. Okay, so the molecular detail of that, what is pertinent to this particular lecture, is something like this: that there are two loci. One is called HML because it is on the left side, the mating type locus L alpha, and HMRA. So on the right hand side, and it is for the A mating type. This is on the left hand side, and it is for the alpha mating type. Now, very interestingly, these cassettes are complete. They have their own promoter, enhancer, every element. But neither of these are actually transcribed. Okay. Now there is an elaborate mechanism through a non-reciprocal, recombination, that this cassette can be transplanted into here, or this cassette can be transplanted into here, copied into this locus. So there is a mating type locus in the somewhere in the towards the center of the chromosome, which is far away from the two ends, the two telomeric ends. That is the active locus. Okay. Now, please understand that these cassettes, either one of these, like depending on how the mating type is switching, this cassette is brought in its entirety from the telomeric locus, near telomeric locus, to somewhat central locus. And then if alpha is brought in here, then it is mating type alpha. If A cassette is brought in here, then it is mating type A. Okay, like do not think that both of these are brought in here simultaneously. It is either this or that. Okay. So now, therefore, the question is that if all the cis elements are there, because as you can imagine, if you just bring it in here, then alpha mating type is imposed. So if all the cis elements are there, and obviously all the trans elements are there, then why is it that until and unless you move it from near telomeric locus to far away locus towards the center, the mating type phenotype is not expressed? The answer is that near the telomere, it is a heavily heterochromatin structure. There are DNA repeat sequences, and the chromatin is very tightly condensed and packed in there, and it does not allow for any transcription to happen either from this or from that. But when you move it from here to here, then it is the same genetic elements, same the transcriptional regulatory elements, but it is no longer in the heterochromatin region, and now the gene is transcribed. It is so interesting that if you artificially clone, introduce the gene for E. coli gene for a methylase, which can methylate a sequence of GATC, then you will see that, so this methylation is absent in yeast. So you will see that because of the presence of the E. coli methylase, many GATC residues throughout the chromosome will be methylated, but not near the telomeric region, including the HML alpha or HMRA loci. What it means is that this region is so compact that even an enzyme like methylase cannot get access to the DNA to methylate it. Therefore, the question of the entire transcriptional apparatus getting entry there and thereby activating transcription is completely out of question. Um, so, so that is what it is. Now, once the cassette is translocated into the central region in the MAT locus, there then, if it is the alpha cassette that is translocated, then alpha 2 and alpha 1 will be transcribed and you will have the alpha mating type. If it is the HMRA cassette that is translocated, then A1 will be transcribed and it will have the A mating type. So this kind of a sequence is therefore referred to as a silencer. Okay, a silencer is a cis element that can silence expression of a gene if like the silence expression of a gene. What it means is that a cassette that can be transcribed somewhere else in the genome, if you put it in the silencer region, it will not be transcribed. Like if you clone any gene in this region, the gene will not be expressed. And this region is generally hypoacetylated. So what happens is that there is a protein called RAP1 that binds to the telomeric region. Once it binds to the telomeric region, it recruits a set of proteins called SER proteins. So SER3 and 4 are recruited, which in turn recruits SER2. And SER2 has a deacetylase activity. So it removes the acetyl group from the lysine residue of histone. Okay, and when you remove the lysine residue of histone, if you remove the acetyl group from the lysine residue of the histone, then what happens is that the acetyl group has a negative charge, lysine is a positive charge. Now, DNA is a negatively charged molecule. So the acetylation neutralizes the positive charge of the lysine residue. Therefore, the interaction between the histone and the DNA is less intense. But once you remove the acetyl group, now the positive charge of the lysine is exposed, and then the interaction between the nucleosome or the histone and the DNA is much stronger. Therefore, it is much more difficult to dislodge it. Okay, and then so therefore that part of the chromatin gets very tightly packed, and because SER2 has been recruited, so it will cause hypoacetylation of some nearby regions. Okay, and then it will also be recruited in the silencer. Okay, so that way this will progress, and slowly more and more regions will be incorporated into the hypoacetylated as hypoacetylated region, and into the heterochromatin region, or whatever you call it. Okay, a silencer or whatever you call it. So therefore, the post-translational modification of a component of histone is actually imparting competence or not onto the DNA that is associated with it. Okay, and it has been shown through many different ways. Like if you do antibody staining, then you can see that if you use a telomere specific antibody staining and the SER proteins, you can see that they are actually co-localized. So you can tell that they are in the same place. And if you mutate this amino acid in the histone in a manner that if you replace that particular lysine with, say, arginine residue, Okay, arginine is also positively charged, but it cannot be acetylated. So then you will not see the any transcription of the alpha or mat HML alpha or HMRA loci. But on the other hand, if you replace it with, say, glutamine, which is neutral charged, or some negatively charged amino acid, then what will happen is that the association between then what is noted is that the haploid cell um, uh, behaves like a diploid. So it cannot undergo mating because it is now expressing both the A pheromone as well as alpha pheromone. So you can tell that it is mating deficient, and just because you changed the histone, like one amino acid in the histone, therefore like the two loci that are nearby the telomere, they are now getting expressed. And so you didn't change anything in the cis element or trans element per se. What you only did is you changed the charge nature of the histone. So this actually tells you that the nucleosome and its assembly with the DNA is very, very critical for transcriptional repression. And what is also known, that there is another kind of repressor, which is called RPD3. RPD3 is also deacetylase. Um, so there is a transcriptional repressor, which is called UME6. UME6 has a DNA binding domain, and it binds to a specific upstream regulatory sequence, which is a little bit away from the Tata box. And to that, UME6 can bind, and its repressor domain in turn can interact with a mediator-like complex containing RPD3 and SYN3, etc. And this RPD3 is a deacetylase, and it imposes repression. And it can be reversed by another sequence called upstream activating sequence, which recruits a transcriptional activator called GCN4, which again has its own DNA binding sequence that recognizes the upstream activating sequence. And the activation domain recruits a complex within which there is one protein called GCN5, which is an acetylase, meaning it adds acetyl group to the lysine residue of the histone. And then the histone gets hyperacetylated, and you get transcriptional activation. So you can the importance of the charge on the histone can be verified using this kind of amino acid substitution in the place of lysine in the histone. Okay, so I hope you got that part. The fact that this compaction, sorry. So there are transcription factors which has activation domain, right? That we talked about earlier. In the olden days, when the amino acid sequence of the activation domains were analyzed, many were found to have a lot of acidic amino acid residues. So there you find in the literature a term called acidic activation domain. This term exists. Now, we will see the importance of activation domain in chromatin decondensation through a very, very interesting experiment. So like before I get there, let me quickly tell you, you have been taught about this Sniff complex, right? Which came from the East genetic screen formatting type switch or sucrose non-fermentation mutation, etc. So the Sniff proteins, many of them, they have kind of DNA helicase-like activity. Okay, uh, and what they do is they kind of push out the nucleosome from the DNA, therefore condensing the DNA. So many activation domain containing transcription factors, many the powerful transcriptional activators, they operate somewhat like this. So this was demonstrated by this, that through homologous recombination, um, lac operator sequence, which is of course not endogenous to yeast, lac operator sequence was introduced into the heterochromatin region of a yeast cell. And then if you artificially express LAC-I, the LAC inhibitor, you can see that it is by doing microscopy using an antibody against LAC-I, you can see that it is located inside the cell, but it is located in a very tight point position, okay? Because the chromatin is very, very tightly bound. Now all you do is, instead of expressing LAC-I, you express LAC-I, which is as a fusion protein to VP16. Now, VP16 is one of the most powerful transcriptional activators known. So when you express the LAC-I VP16, then you suddenly see that this fusion protein, which can be detected by the same antibody, is now distributed in the nucleus all over the place. So this tight heterochromatin now has decondensed, and you can locate it. So what the transcriptional activator is doing is it is basically acting by decondensing the chromatin. So what was erstwhile a heterochromatin region, just because you artificially recruited a transcriptional activator like VP16 there, it acted by decondensing the chromatin. Okay, so that's how these transcriptional activators and repressors work. We have already talked about the mediator, and therefore I am not getting in the discussion all over again. So now we quickly move on to another class of transcription factors, which I have referred to earlier, which are called the nuclear hormone receptor transcription factors. The nuclear hormone receptors, they are basically transcription factors. They bind to these small organic molecules, which are called nuclear hormones. And there are various types of nuclear hormone receptors known. Estrogen receptor, progesterone receptor, glucocorticoid receptor, thyroxine receptor, etc., etc. Notice very interesting thing. The DNA binding domain for all of these receptors, they're very similar to each other. The amino acid identity is roughly between 42 to 94%. Okay. The ligand binding domain, obviously the ligands are very different from each other. So there the identity is relatively less, much less. Okay. But the activation domain varies completely from each other. Okay. So you can understand that what might have happened is that there was an original, the founder nuclear hormone receptor, and that molecule somehow got duplicated, and therefore the DNA binding domain remained the same, but with time it acquired specialized property for binding different kinds of nuclear hormones. And obviously the activation domain are very different from each other. And these are, we're talking about millions of years of experiments, and eventually what we have this diverse array of nuclear hormone receptors. But all of them, if you look at their binding sites, the DNA binding domain, they have some kind of similarity in them. You see, retinoic acid receptor, thyroid responsive, This is retinoic acid response element, okay, RARE, retinoic acid response element, thyroid response element, vitamin D response element, estrogen response element, glucocorticoid. So if you look at it, you see that this particular sequence, AGGTCA, that is there in all of these. Okay. Now, in some cases, there is a direct repeat of that. In between, you have either 3 nucleotide or 4 nucleotide or 5 nucleotide. So depending on that, it will bind to vitamin D or the thyroxine or retinoic acid. For estrogen response element, it is still AGGTCA, but there is a inverted repeat. Okay, and glucocorticoid receptor is like slightly different from this signature, but you see that how similar they are to each other vis-a-vis the sequence to which the nuclear hormone receptor will bind, and therefore it does not come to you as a surprise that the nuclear hormone receptor DNA binding domain is very similar to each other, right? And all of them has the same property that once it binds to the ligand, it moves into the nucleus. And this is very simply shown that, with glucocorticoid receptor. Now, glucocorticoid receptor normally is in the cytosol. When you, okay, so how it is done? That is like beta galactosidase. It has nothing to do with nuclear translocation or anything like that. It is expressed in a mammalian cell, and whether it is with or without an agonist for glucocorticoid receptor, dexamethasone, it stays outside in the cytosol. Okay. Now, what you do is you fuse on the N-terminus the glucocorticoid receptor to this beta galactosidase. And when you do that, then you can see that in the absence of dexamethasone, it is still in the cytosol, but in the presence of dexamethasone, it has all moved into the nucleus. Okay? And you do not need the DNA binding domain for that. So if you eliminate this part, the DNA binding domain or the transactivation domain, you only keep the ligand binding domain. Even then, it moves. So this is an independent activity for that particular. So what you can imagine, perhaps, like I have not studied, I have not gotten into the depth of it. I'm just imagining that what is likely to be happening is that this ligand binding domain has a structure that is such that once it binds to the ligand, that is estrogen or what dexamethasone or whatever it is, then a pseudo nuclear localization signal gets opened up, and the transcription factor moves to the nucleus. So now this is a biological phenomena, so that the biological response is there only when the nuclear hormone receptor is there in the cell. And that's how it responds to systemic changes in the hormonal level. And if you remember, this is exactly the property that has been utilized when when scientists developed this conditional knockout. Okay, that the Cre recombinase is fused with a mutant version of estrogen receptor, which has a much lower affinity towards natural estrogen, but very high affinity towards an analog of estrogen called tamoxifen. So when it binds to tamoxifen, instead of dexamethasone, you just think tamoxifen. So minus tamoxifen, Cre is outside of the nucleus. Plus tamoxifen, Cre moves in, and recombination happens. So that's how it is. So that's all that we had to learn in transcriptional regulation. We will now start with a new chapter, post-transcriptional regulation, pretty soon. Okay, thanks.